Good morning. My name is Dr. P. Mutterna. Today we would like to discuss on adenovirus. What is adenovirus? We all know that adenovirus is a non-enveloped icosahedra shaped double-stranded DNA containing humans and as well as animals infected virus molecule. It causes upper respiratory infections, and also sometimes it also causes conjunctival gastrointestinal infections. When you see the properties of adenovirus, as I said, it is a non-enveloped icosahedral shaped virus. Roughly it has length up to 65 to 80 nanometers, and also it looks like in the form of satellite shape like structure. Inside that virus you do have a linear double-stranded DNA with the length of up to 30 to 38 kilobase pairs of linear DNA molecule. And at the ends of that linear DNA molecules, at the ends of the linear double-stranded DNA molecules, that viral DNA towards the 5th prime, it is attached with terminal protein. So this terminal protein, it is involved in the replication process. After replication, it encodes roughly 30 to 35 protein molecules. In that 15 protein molecules, we used to call it as viral protein molecules. They are involved in the building of that virion structure. And both strands of the DNA which are present in that virus, when that virus enters into the cell, the host cell, both strands are get transcribed into functional mRNA molecules. The transcription and as well as the replication occurs inside the nucleus of the cell, and the gene expression of these viral proteins occurred in the manner of timely expression. So they get expressed in the form of order-like structure. And the assembly of the virus, after synthesizing their own proteins and as well as DNA molecules, viral double-stranded DNA molecules, the assembly occurs inside the host nucleus. As I said, it will cause upper respiratory infections because it will get transmitted through the aerosols or droplet nuclei, one person to the another person through the droplet nuclei. Due to that, it causes the upper respiratory infections. Because of that infections, there will be a secondary infections in the form of bacterial pneumonia, and also it will cause some kind of what is called gastrointestinal infections and also conjunctival infections in the eye. There is a special property of this virus. Once you infect this virus, particularly in the animals, it induces the what is called tumor formation. It induces the tumors inside the rodents. Nothing but it causes the cancers or tumor formations inside the rodent animals. What about the transmission? It can be transmitted through what is called ingestion or fecal-oral route. Sometimes it will also get transmitted through droplet nuclei. As we have been seeing, the transmission is similar to the coronavirus, the COVID-19 coronavirus, because it will get transmitted from one person to the another person through the droplet nuclei, and also it will get contaminated or it will get transmitted through contact lenses or from hand to eye, from handshaking from one person to the another person. What is the structure of adenovirus? We all know that our adenovirus looks like in the form of what is called a short-light shape like structure. Basically, it is a hexagonal icosahedra shape like structure. We all know that benzene, the structure, nothing but hexagon. So in that hexagon, it has different types of viral proteins. They are filled with the each type of capsomere. They are filled with the each different types of shapes of capsomeres. Then due to that building, they try to form the viral capsid. And also they do have the knobs. These knobs are nothing but a glycoprotein like structures. They are involved in the attachment to the host cells. And the first one we used to call it as a knob. Then next one is a penton and a exon. So you have three types of proteins. We can call it as viral proteins. They are involved in the building of the adenovirus structure. One is called knob protein, another one is called penton base, then followed by the exon. Then the remaining portion, the knob, it is attached with the penton base with the help of a fiber. Then inside the virion, we can also call it as inside the core, you have the double-stranded DNA. When you see in detailed picture of this adenovirus, you have three set of protein molecules with glycoprotein spikes, and also it is a non-enveloped icosahedra shaped structure. The three set of proteins are capsid, scaffolding proteins, and also DNA binding proteins. So inside the capsid you have hexagon polypeptide 3 and nothing but penton base, then followed by the a trimeric polypeptide 4 fiber proteins. So the capsid is made up of exons, pentons, then followed by the fiber. Then the fiber is attached with the nab. Then the other proteins are called scaffolding proteins. They are nothing but monomers of polypeptide of 6, monomers of polypeptides of 9. And also you have other proteins in the form of 9, 3A and 6. They forms the scaffolds that holds the capsid together. So scaffolding proteins are nothing but 6th, 9th and also 3. They are involved in the building of the scaffolding. Then inside the core you have the DNA binding proteins in the form of protein 5th, protein 7th and also terminal protein, and also you have what is called protease enzyme. So these DNA binding proteins, they are involved in the replication purpose and also they are involved in the transport of the DNA. Genome. We all know that the genome of adenovirus, it is a double-stranded like structure, that too in the form of linear like structure. The genome of adenovirus towards the 3 prime, it is attached with ITRs, internal terminal repeats. ITRs are nothing but internal terminal repeats. So those internal terminal repeats are attached towards the 3 prime end of that double-stranded DNA. And also this internal terminal repeats, they will be acting as a ORIC, nothing but ORI regions, nothing but origin of replication. So the replication will be start from those ITRs. And towards the 5th prime, you have the terminal protein. This terminal protein, it will be acting as a primer for the initiation of replication. We all know that roughly The length of this double-stranded DNA molecule from 32 to 38 kilo base pairs. Life cycle. In order to understand the viral infection of this adenovirus, we have different steps in the life cycle. The first one we used to call as attachment, penetration, transport, transcription, then followed by replication, then followed by the assembly and release. Initially, this virus, after transmission, it binds to the host cell, particularly upper respiratory cells or inside the intestinal cells. Then after that, it will undergoing for the penetration. Then after penetration, once it reaches to the cytoplasm, from the cytoplasm it goes to the nucleus where it delivers its double-stranded DNA. Then that viral DNA, once it reaches to the nucleus, it starts the transcription, then followed by replication. By using those three types of gene expression process, then it is involved in the synthesis of viral proteins and also viral enzymes. Then it goes for assembly. The assembly occurs inside the nucleus. Then after that, it will come out from the nucleus and it reaches to the cytoplasm. From that, it will come out into the outside the cell. So that is known as release. First one is attachment. Our adenovirus, it has a knob-like structure, nothing but glycoprotein spike. So by using that glycoprotein spike, it goes for the attachment. On the surface of the epithelial cell, for example, this is our epithelial surface membrane, plasma membrane. On the surface of epithelial plasma membrane, you have different types of glycoprotein molecules. One is called integrin, another one is called CAR. CAR means Coxsackie adenovirus receptor. By using those two receptors, our adenovirus try to bind to this receptor. As I said, on the surface of host cell, you have a plasma membrane. On the surface of the plasma membrane, you have different types of proteins. Those are nothing but glycoproteins. Those glycoproteins, they are involved in the cell-to-cell interactions. But unfortunately, those glycoproteins will be acting as a receptor site for the adenovirus. Those are CAR and integrin. Integrin, we all know that it is involved in the cell-to-cell interaction. CAR means Coxsackie adenovirus receptor. Those two receptors, they are acting as a attachment site for the adenovirus. So that adenovirus, slowly it will come towards the plasma membrane of epithelial cell, which is present inside the respiratory tract or inside the gastrointestinal tract. With the help of electrostatic interactions, there is some kind of interaction between the integrin and as well as the knob-like structure. First, it binds to the integrin with the help of knob-like structure. Then slowly it moves towards the CAR protein, Coxsackie adenovirus receptor. So the CAR will be acting as a receptor site for the adenovirus and integrin will be acting as a co-receptor. Then because of electrostatic interactions, this virus slowly it will get embedded inside the two CAR proteins, just like a lock and key mechanism. Once it will get integrated, once it bound to the CAR-like proteins, then there will be internal signaling. Due to that, there is a actin polymerization below the plasma membrane. Due to that, there is a polymerization of actin filaments. Due to that, they slowly try to form a pit-like structure. This pit is nothing but a clathrin coated pit. We can also call clathrin associated endosome formation. Then because of that, they try to form the endosome. That concept is known as penetration. So once it forms the endosome, slowly it will get inside. So this is known as endosome formation, and the entire process from here to here we can call it as penetration, and this one we can call it as attachment. Once the endosome is formed with the help of actin filaments and clathrin coated pit, clathrin proteins, the endosome slowly moves towards the nucleus. That is known as transport. So our adenovirus needs to get transported from upper cytoplasmic space to the inner cytoplasmic space, nothing but nearer to the nucleus. During that transport, there is a degrees of hydrogen ion concentrations inside the endosome. Due to that, there is a destabilization of the core proteins which are present inside the adenovirus, nothing but scaffolding proteins. Then slowly it delivers the core of the virion, and this core of the virion will get transported nearer to the nucleus. This core, once it is delivered inside the cytoplasm, it needs to get transported towards the nucleus with the help of microtubules and also dynein proteins. With the help of dynein proteins, with the help of microtubules, this core of the adenovirus will get transported nearer to the nucleus, where this adenovirus core it binds to the nuclear pore and it delivers the double-stranded DNA into the nucleus. This is known as transport. Once it enters into the nucleus, it starts the transcription. It starts the transcription, nothing but gene expression. In adenovirus transcription, it expresses 5 early and 1 late mRNA molecules. The early mRNA molecules, they are involved in the early mRNA molecules, they are modified by the differential splicing process. Due to that, you get E1 to E4 proteins, and the late mRNA molecules, they are produced from single transcript of 29,000 nucleotides, and also that get spliced and differentially polyadenylated to yield 18 different types of mRNAs. And this, because of that splicing mechanism, adenovirus will be acting as a model virus for the studying the mRNA splicing in eukaryotic organisms. It will be acting as a model organism. So it will be acting as a model virus. So what are the functions about the early gene products or early gene expression? Basically, the early gene expressed proteins, they do have 3 functions. First one, they induce the host cell to enter into the S phase, particularly E1A and E1B. So E1A and E1B, they try to bind to the retinoblastoma protein and also P53 protein. Retinoblastoma protein, RB protein and P53 protein, they are involved in the controlling of the cell cycle, nothing but regulation of the cell cycle. But E1A and E1B, they try to bind to those two proteins. Due to that, the cells directly they can enter into the S phase. The cells directly they move from G1 to the S phase quickly. Nothing but the checkpoints which are present in the cell cycle, they could not control the cell regulation. That's why they try to produce the tumors. Apart from that, they are also responsible for the transformation, and also they will be acting as the oncogenes. They causes the oncogenesis, nothing but tumors formation. And also the proteins which are coming from early gene expression, they protect the virus from the host defensive mechanism. And also they synthesize replication associated protein molecules. Some kind of proteins which are coming from the early gene expression, they are involved in the replication of the viral DNA. As you can see here, we have even a protein that specifically binds to the RB. Due to that, the RB will be phosphorylated. Because of in the phosphorylation form, it cannot bind to the E2F protein. Then the E2F protein is free. The E2F protein is free. This E2F protein involved in the movement of G1 phase cells into S phase. So quickly all the cells which are present in the G1, they goes into the S phase. Similar way, E1B protein, it binds to the P53. Due to that, it inhibits the apoptosis of viral infected cells. It inhibits the apoptosis of viral infected cell. So those are the two functions. So because of that, the adenovirus can able to produce the tumors inside the animals. What about the replication? We all know that it is a double-stranded DNA. With the help of terminal protein, the terminal protein will be acting as a primer for the virally synthesized DNA polymerase. There is a synthesis of viral proteins. One of the protein will be acting as a DNA polymerase. So that DNA polymerase bind to the TP protein, then it initiates the replication process. This replication process also called strand displacement replication. It is similar to the Okazaki fragments, but here we can call it as strand displacement type of replication process. So there is a synthesis of new nucleotides by using other strand as a template strand. So one strand will get displacement, then the other strand will be acting as a template, then there is synthesis of new strand with the help of virally synthesized DNA polymerase. Then once the one single strand synthesis over, then it try to form circle-like structure, a pan-shape-like structure. Then it starts synthesizing the another opposite strand. This entire process is known as strand displacement mechanism. That is about the replication. Now, once again, I will repeat the concept of life cycle of adenovirus. We all know that initially, adenovirus, it binds to the integrins and as well as CAR proteins. With the help of those host proteins, there is a formation of clathrin coated vesicle. Due to that, it forms a endosome through the process of clathrin mediated endocytosis. Then that endosome, or we can also call it as vesicle, it will get transported nearer to the nucleus. During that transport, there is a decreasing of pH inside the endosome. Due to that, it delivers the core, and once it reaches to the site of the nuclear pore, the core binds to the nuclear pore, then it delivers the linear double-stranded DNA into the nucleus. So that linear double-stranded DNA, we can also call it as a mini chromosome. By using host metabolic enzymes, initially it will undergo for transcription. Initially it synthesizes different kind of protein molecules. Those protein molecules we can call it as immediate gene expression and also early gene expression. Then once it is over, then it goes for what is called DNA replication, that is known as strand displacement replication. During that also it synthesizes intermediate protein molecules through the intermediate gene expression. Then after that there is a viral assembly inside the nucleus. Then it will come out from the nuclear pore, then it reaches to the nearer to the plasma membrane. With the help of protease enzyme, it cleaves the plasma membrane, then it will come out from the cytoplasm as a matured virion. Then it infects the nearby cell. This is the life cycle of adenovirus. I hope you have understood well. See you in the next class. Thank you very much.